Hello everyone and welcome back to our channel Biodiversity and Conservation. I am Subhalakshmi Raut from Ambika Prasad Research Foundation, Odisha, India. And today we are going to study about ligand and receptors. So this is a common diagram showing ligand binding with receptor and how the process of cell signaling is carried out. Ligand first binds the receptor, activating it, which causes some reactions in the target cell, and it gets activated. There are some conformational changes also after the ligand binds with the receptor and forms receptor ligand complex. Then after these changes, there is initiation of signal transduction, and then the processes continue of cell signaling. So first of all, ligands. What are ligands? Ligands are any signaling molecules, and they are chemically heterogeneous compounds, and they are mainly divided into two categories, that is membrane bound and other one is secretory signal molecules. Membrane bound signal molecules remain bound to the surface of the cells, and they communicate or mediate through contact dependent signaling. And in the previous cell signaling slide, we studied that contact dependent signaling are mainly juxtacrine signaling, and in most cases signal molecules are secreted by signaling cells, and they are called secretory signal molecules. The secreted extracellular signal molecules are then further divided into three general categories based on the distance over which the signals are transmitted. They are endocrine signal molecules, paracrine signal molecules, and autocrine signal molecules. The extracellular signal molecules are synthesized and released by signaling cells and produce a specific response only in target cells, and that have either cell surface receptors or intracellular receptors for the signaling molecules. And if we Next, we'll study about the extracellular signal molecules, which are broadly divided into small lipophilic molecules and hydrophilic molecules. The small lipophilic molecules are lipid loving, so they can diffuse across the plasma membrane and interact with the intracellular receptors, that is, the cytosolic or nuclear receptors inside the cell. And hydrophobic molecules, they hydrophilic molecules, they are um, lipophobic, or they cannot pass through the lipid bilayer plasma membrane without the help of any receptors. So it will bind to cell surface receptors, so that can pass its signal from to the receptors present inside the cell membrane. And few lipophilic signal molecules, they also bind to the cell surface receptors, and most of these molecules are members of eicosanoids, which include prostaglandins, prostacyclin, thromboxanes, and leukotrienes. And all the eicosanoids are synthesized from arachidotic acid, which is formed from phospholipids. However, most of the extracellular signal molecules are hydrophilic, and they bind to the cell surface receptors of the target cell. There are some examples of signal molecules that interact with cell surface receptors, that is hydrophilic molecules. Examples are epinephrine, norepinephrine, glucagon, insulin, um, gastrin, secretin, cholecystokinin, and ACTH, adrenocorticoid thyroid hormone. Uh, the uh, there are some examples of signal molecules or hormones that interact with the intracellular receptors, that is cytosolic cortical receptors. Like they are of three categories: steroid hormones, which include progesterone, estradiol, testosterone, cortisol, corticosterone, aldosterone. And the second category is steroid-like hormone like alpha-ecdysone. And the third category is non-steroid hormones, which include thyroid hormone and retinoic acid. This is the diagram which shows how hydrophobic and hydrophilic signal molecules transfer through the cell membrane, and how signal transduction occurs here. First, the hydrophobic molecules, that is lipophilic molecules like steroids, retinoids, and thyroxine, they are hydrophobic means lipophilic, and lipophilic means lipid loving, and the plasma membrane is formed of lipid bilayer, and so they can pass through the plasma membrane easily, and they pass through the plasma membrane and bind with the cytosolic receptor, um, that is a type of intracellular receptor. They bind to the intracellular receptor and go through the nuclear membrane, or it goes directly into the nuclear membrane and binds with the nuclear receptors, and take part in in the pathways of gene expression or development of body. And there are hydrophilic signals like small molecules of peptides, proteins, or like I said, epinephrine, nephrin, glucagon, insulin, they all require cell surface receptors to pass through the cell membrane. And generally, the cell surface receptors remain inactive on the plasma membrane, but after binding with the ligand, it gets activated and forms ligand receptor complex, which then um, produces cascade reactions. That is, it goes through some steps, like after binding with the ligand, the receptor activates and gives signal to another receptor get activated, or forms a product which will bind with another receptor to get activated. So in the fifth to sixth steps, it shows the different signal transduction proteins and secondary messengers, and then it will activate the effector protein, which will mainly give the reaction for modification in cellular metabolism or in gene expression or in development, all that processes. So binding of extracellular signaling molecules to the cell surface receptors leads to an increase or decrease in the concentration of low molecular weight intracellular signaling molecules, called secondary messengers. Some secondary messengers are cyclic AMP, cyclic GMP, DAG, which is diacyl uh, glycerol, IP3, that is inositol 1,4,5 triphosphate, phosphoinositides, and calcium. They are main secondary messengers that are seen in the process of cell signaling in our body. Now, what are receptors? Receptors are mainly chemically protein or glycoprotein molecules that bind to the signaling molecules, that is ligands. The cellular response to a particular extracellular signal molecule depends on its binding to a specific receptor located on the surface of a target cell or in its nucleus or cytosol. The binding of a ligand to its receptor causes a conformational change in the receptor that initiates a sequence of reactions leading to a specific cellular response. And on the basis of location, receptors are mainly divided into two categories, that is intracellular receptors and cell surface receptors. We will go through one by one. Intracellular receptors are those proteins that are located in the cytosol or the nucleus. These include receptors for the steroid hormones, thyroid hormones, retinoids, and vitamin D, as well as some different orphan receptors. The intracellular receptors are all structurally related and belong to nuclear receptor superfamily. Within a cell, the intracellular receptor ligand complex controls the activities of the responsive genes, and a large number of nuclear receptors have been identified through the sequence similar to the receptors, but they are not identified, they do not have identified natural ligand, so referred to as nuclear orphan receptor. So nuclear receptors are a family of highly conserved transcriptional factors that regulate transcription in response to small lipophilic signaling molecules. And the members of the nuclear receptor superfamily can both positively and negatively regulate transcription. And most members of the nuclear receptor superfamily consist of a N-terminal activation domain, then a central DNA binding domain, and a C-terminal ligand binding domain. And in case of steroid hormones, the receptors are located in cytosol, and in the cytosol the receptors remain in inactive form, and after making complex with heat shock protein HSP, and the ligand binding with the receptor in cytosol results in the dissociation of HSPs and translocation from cytosol into the cell nucleus. Once the ligand receptor complex is in the nucleus, the DNA binding domain of the receptor binds to specific hormone response element of the DNA, and in this case actually the steroid receptors bind as homodimers to inverted repeats spaced by 3 nucleotide spacer sequence. A hormone response element HRE is a specific DNA sequence that a receptor recognizes with high affinity, and it binds to the steroid receptors, which then causes cascade of reactions, and we will see gene expression changes in or modification in the gene expression. But unlike steroid hormone receptor proteins, the thyroid hormone receptor proteins are located in the nucleus bound to DNA, and even in the absence of their thyroid hormone ligand. The thyroid hormone response element of DNA has binds to only one of the half side because it has two half sides, and the other DNA half side binds to retinoid X receptor RXR. When the thyroid hormone receptor and RXR bind to two DNA half sides of the hormone response element, the two receptors form a heterodimer So in case of steroid hormone, it forms homodimer, and in case of thyroid hormone, it forms heterodimer. So these are intracellular receptors. Next is cell surface receptors. Um, they are also known as transmembrane receptors, and are cell surface membrane anchored proteins that bind to external ligand molecules. This type of receptor spans the plasma membrane and performs signal transduction through which an extracellular signal activates intracellular signal. The ligands that interact with the cell surface receptors do not have to enter the cell that they affect, and some cell surface receptors are also called cell specific proteins or markers because they are specific to individual cell types. And all the hydrophilic signaling molecules as well as some lipophilic molecules bind to the cell surface receptor proteins, and these cell surface receptor proteins act as signal transducers to produce intracellular messengers. These messengers stimulate the sensors and effectors responsible for activating cellular processes. So mainly there are three classes of cell surface receptors. That is, first one is ion channel linked receptors. Next one is enzyme linked receptors, and third one is G protein coupled receptors. We will study one by one in the next slides. So here the cell surface receptor is mainly present on the surface of the cell, and it is very particular on its ligand. It will bind with the particular type of ligand which is required for the process of body, and so it can be said as lock and key mechanism because a particular key can only open a particular lock. So each cell surface receptors has three main components: an external ligand binding domain called the extracellular domain, a hydrophobic membrane spanning region called a transmembrane domain, and an intracellular domain inside the cell that can connect to a specific signal transduction pathway. The size and extent of each of these domains vary widely depending on the type of receptor. So this is the summary of how many types of receptors we have. Um, there is intracellular receptor, there are cell surface receptor, which are of three types: ligand gated, G protein coupled, and enzyme linked receptor. Here it's shown in very short how ligand comes from extracellular fluid into the nucleus and binds with the receptor. Here the receptor is intracellular receptor. It gives some signals, and which passes through a cascade of reactions, and we get modification or changes in the gene transcription according to the ligand binding, and it carries out protein synthesis, and we see the cellular effects. And in case of ligand gated ion channel, here after the ligand binds with the receptor, it opens the ion channel, which helps the influx of ions from extracellular fluid into the cell, where we can see hyperpolarization or depolarization, that is creating an action potential. It's mainly seen in skeletal muscle contraction, and we can see the cellular effects here. And the G protein coupled receptor is a very particular type of cell surface receptor, and it's a very intricate receptor complex that forms with ligand. After the ligand binds with receptor, it gets phosphorylated or gets activated, and then it can go either way. It can activate the ion channels to um, facilitate the passage of ions from extracellular fluid into the cell, which changes the um, ionic concentration of the cell, and we can see the cellular effects. And in there is other way also in which G protein coupled receptor acts. When the G protein gets activated, it binds or it activates another, if it activates the effector, which activates secondary messengers. And the secondary messengers act in three ways. That is, it releases calcium, which will go through some cascade reactions to produce the required cellular response. And it can also, the secondary messengers can also activate protein phosphorylation, which will also give us the cellular response. And in other ways also it can help to get us the cellular response. And the third type of cell surface receptor, enzyme linked receptor. Here the ligand binds with the receptor, which is actually an enzyme. And mainly uh, all the cellular reactions where enzymes are included, this type of reaction can be seen. It's common. And after binding with the ligand, the receptor gets activated and gives protein phosphorylation occurs, and then which affects the gene transcription, and then there will be changes in protein synthesis, and we will see the cellular response. So this is all from me today. Thank you.